Okay, so it's great to be here. I'm very pleased to be here in San Diego. Last time I've been here was 23 years ago. And I'm going to introduce you today our new covalent FPDD approach that we have established. In Wuxi biology, one of the main topic is supporting many of our partners in developing new drugs. And we have different hit generation technologies like HDS, DEL screening, fragment screening, and virtual screening. Depends on the target, on our experience. We decide which technology or which screening method should be used. Exactly. So, but now I would like to focus a lot on fragment-based drug discovery. We have two different libraries. Non-covalent library that contains about 3,000 fragments that we're using now already for 4 to 5 years very successfully. And this year we have introduced a covalent fragment screening with different warheads that I'd like to discuss with you today. And as you know, if we want to run a FBDD project, what we need is a protein with a very high quality. So we have a relative big protein production unit that support us. And of course, in order to run FBDD project, robust biophysical technologies are very important. So for non-covalent fragment screening, we mainly use SPARE and MST, while for covalent fragment screening, mass spec, of course, is typically the method of choice. Nevertheless, we have run already covalent fragment screening by MST in the past pretty successfully, but mass spec is a more direct method. And what is, of course, very important in order to be successful in an FPDD project, structural biology is enormously important, since we can initiate a medchem and fragment evolution only when we can combine biophysical data together with structural biology. And coming back to the protein, so everything starts and ends with the protein quality. So we have a very big team in Germany and in China, very experienced in protein production that support us a lot. And they made over the years protein, very challenging protein as well, belonging to very different protein classes. Can be helicases, can be immunomodulator, can be membrane protein, His ligases, and so on. But for the screening, what we need is robust biophysical assay. So again, for covalent fragment screening, we are using a rapid fire and so high throughput mass spec, while for non-covalent fragment screening, MST trick and SPARE are our method of choice. Depends on the target. During assay setup, we test different methods and then decide which technology should be used for screening. Other important technology are, of course, nano-DSF, DLS, and BLI depends on what needs to be measured. So coming back to the library, so the library was actually finalized about 2 weeks ago. So it's a very new covalent fragment collection. The unique things about it, it was completely designed and synthesized in Wuxi. So it's not a commercial available compound from other provider like Hanamin and others. Our medchem team have designed the library out from 15,000 building blocks, show good diversity and with different warheads allowing to target cysteines and lysines. So regarding the workflow, I don't think you will see any very big surprise. So primary screening would be typically using mass spec. In some cases, we can consider to do MST. We did MST as a covalent fragment screening technology a lot in the past, as we didn't add a rapid fire. But now when we have higher throughput, I would typically initiate the screening with a rapid fire, run orthogonal assay with the best hits. Typical hit rate are about 5%. Orthogonal assay can be, for example, MST nano-DSF. In case of an enzyme, we can run a functional biochemical assay like an ADP glow, and the biochemically validated hits would be go into crystallography, which is, of course, for covalent compound very important as well. And typically, I would say a compound number that we can deal very well is about 50 compounds. And then combining structural information again with biophysical, biochemical data will allow us to initiate heat expansion. So today I'd like to share with you a case study. So the first case study that we did was BTK. BTK is an important regulator of cell proliferation and cell survival. And as we saw already in one of the talk today, and we know BTK is a very nice target to get started since the kinase domain of BTK harbor 2 solvent export cysteines, and so acrylamide is the right way to go for BTK, and there are very well characterized covalent inhibitors like ibrutinib. In order to set up the assay, our medchem teams have established tool fragments for us based on the known structures of the covalent inhibitors, and that was very useful in order to establish the assay. So for the screening, what we did, we have just screened a subset of the library, only the acrylamide part. So we have screened about 200 covalent fragments, acrylamide at concentration of 10 micromolar and 1 micromolar protein. We incubate the sample for 6 hours and measured. Heat rate was pretty high, pretty much expected for the acrylamides. 62 binders out of 200, so that's about 30%. And then we have clustered the hits into 3 different groups: complete covalent binders, as we can see on the left, partial covalent binder in the middle, and just very weak covalent binder. Then we did our first orthogonal validation. So we have run in nano-DSF. In nano-DSF, we typically see it as a very good orthogonal method for mass spec, and as you can see it here, it was working great. All complete modifier have increased BTK thermal stability between 2 and 7 degrees. So that was really very, very nice readout. And in order to get an idea about the potency of our covalent fragments, what we did, we have established an MST experiment with ibrutinib, which gave us a KD of about 1 nanomolar. And then we've measured whole compounds after relative short incubation time of 30 minutes. And we observed for all compounds that we identified in mass spec as a complete binder, reasonable, I would call it aberrant KD in the low micromolar range, which we need to keep in mind that's an aberrant KD. It's not a real KD. It just gives us an idea about the potency after 30 minutes. But since we know that for covalent compound consists of binding, which take place, and then the reactivity of the warheads, we have run the MST experiment for the best binders at different time points. And you can see beautifully how the binding affinity is increasing during 6 hours incubation time. So you can see that directly after mixing, we have measured KDs in the low micromolar range for this compound, and after 6 hours, we have measured aberrant KD of 30 nanomolar. And since we have observed relative high potency, we have run ADP glow assay for the best binders, and we see again very nice correlation between the MST data, the mass spec, and the nano-DSF, and we were able to demonstrate that the fragment can very nicely inhibit the kinase activity of BTK. And then after we have our biochemically, biophysically validated hits, we went back to our medchem team. Then they ran some talking where we have able to see a conserved region that our fragment, similar as ibrutinib, bind or most likely bind to the hinge region of BTK and covalently modified cysteine 481 of the protein. And we were very happy that we were able to solve structures with a resolution of 2.6 Armstrong, where we see again very nicely the covalent modification of cysteine 4H1. And we can see from the binding pocket that the MedChem team can now play a bit more in order to get it a bit, fill the binding site and going to get a more potent and more selective BTK inhibitor. So that's the plan for the next. So this is really the project was started actually 1.5 months ago. We just finished it. It's just an update that the last data was generated about 2 weeks ago. So we are still kind of in the middle. And as you can see it here, we are initiating now fragment evolution by MedChem, and we plan to synthesize 10 to 15 compounds. This is just an example of, we would consider 3-4 iterative cycles of fragment growth, and we will test, of course, every compound in nano-DSF MST mass spec and crystallography, and we hope to be able to improve the potency and the selectivity. So the next plan, again, we have few very interesting other compounds, so we still wait to get few more structures, fragment evolution, and we have established very nice on MST for BTK a target engagement assay where we measure the binding affinity in MST directly in eukaryotic cell lysate, and that will allow us to have ideas about target engagement as well. And that's all. I hope you got a good impression about how we run a covalent fragment screening in WuXi, and I'm happy to take few questions, and I'm still here till the afternoon if someone wants to discuss more.
Can you hear me?
Yes.
That's really cool stuff you guys have it. One question I have in the terms of the practical aspect, when we try to that the early space of discovery and the validation part, and like Catch-22, you don't have the assays and you have a fragment or virtual screening. You follow up at the nano-DSF and MST. But historically, we have seen it that there's no consistency between the nano-DSF and MST. When you generated the data of those two particular assays and you try to see the justify, they are everywhere. Have you done any systematic studies and the case studies which you can guide to the other audience and the people when they come back again and do that studies?
So we need to consider what are nano-DSF and what are MST. So in nano-DSF, what we are measuring how compounds affect protein terminal stability. For some target, depends where the compound binds, you will see very strong impact, but depends where the compound bind, they will not show any impact in nano-DSF. So what we see in our lab, and we have established about 250 different protein targets in the last years with MST and S-pair, we see very good correlation between nano-DSF and MST for all stabilizers. In case of destabilizers, many cases it's difficult. It's thermodynamically even different to explain why protein decrease thermal stability at all. Therefore, destabilizer is the one that typically I kick out of my collection. I know there's other people that see it a bit differently, but that's at least how I see it, and we run many. We want to see stabilizer, and for covalent compound, you typically see very significant increase in protein thermal stability. KRAS, for example, with the Amgen compound, the covalent inhibitor stabilized 25 degrees. And this is pretty well accepted. And we see a relative good correlation, I would say, between nano-DSF and other biophysical methods.
I think in the interest of time, let's save further questions for later, and thank Moran and the rest of the speakers for really stimulating session this morning.
Thank you so much. Dessert break with posters in the exhibit hall, plus a raffle at 1:20.